Welcome everyone, and thank you very much for joining us. My name is Alex. I'm the Chief Business Officer at IOTA Sciences, and I will be moderating today's webinar. Recent progress in the culture of induced pluripotent stem cells, coupled with ever more powerful genome engineering technologies, continues to advance and transform our understanding of the genetic and molecular basis of human disease. However, to fully exploit the potential of these technologies, scientists still need to overcome several bottlenecks that remain throughout the respective experimental workflows. In today's session, we will hear from two experts on how they continue to streamline and automate processes for successful genome engineering in pluripotent stem cells. Dr. Narasimha Telugu, our first speaker, holds a PhD degree from the University of Cologne, Germany, where he used CRISPR technology and induced pluripotent stem cells to unravel the role of microRNAs in the proliferation of cardiomyocytes. Narasimha then joined the pluripotent stem cell technology platform at the Max Delbrück Center in Berlin, Germany, where he now manages genome engineering projects of iPSCs and focuses on the development of simple, efficient, and streamlined genome engineering processes for iPSCs. Our second speaker today is Dr. Katja Mattis. Katja holds a PhD degree from the University of Oxford, UK, and is a research scientist at IOTA Sciences. She supports our customers with everything related to single-cell cloning and stem cells. She has several years of in-depth knowledge and expertise in genome engineering and induced pluripotent stem cells across academia and the biotech industry. Now, before we get started with the actual presentations, just a little bit of housekeeping up front. At the end of the talks, we will open a question and answer session. And if you'd like to ask a question, either during the talks or at the end of the talks, please type your question into the Q&A box in your control panel, which you find at the bottom of your screen. And now, without any further ado, I'd like to hand over to our first speaker, Narasimha Telugu.
Thank you, Alex, for the nice introduction, and hello to everyone. I am Narasimha, and today I will share my experience of genome engineering in human-induced pluripotent stem cells, commonly known as HIPS, and from now on, I abbreviate them as human iPS. Before deep dive into the process of genome engineering in human iPS, I would like to introduce the contents of my talks. Firstly, I will speak about the potential applications of CRISPR editing in iPS. Next, I will give you a brief overview about the available genome engineering tools and their role in different types of genome modifications. Finally, I will introduce the assured process to make successful engineering in human iPS using CRISPR-Cas9 with an example of making functional knockout in human iPS and correction of heterozygous disease-specific SNP in human iPS. As you all know, the advent of large-scale sequencing technologies with the affordable price, we have been learning that role of many genetic variants in human diseases. There are 7,000 types of rare genetic diseases, and 1 in every 17 people is having genetic related disease in one point of their life. Moreover, 80% of these diseases are with no treatment. Therefore, it is important and necessary to understand the genotypic link to disease phenotype. With the combination of iPS and CRISPR technology, we can establish a definitive phenotype to genotype to phenotype links that would help us to develop new drugs or new gene and cell-based therapeutics. And I assume most of you know about what is iPS technology. And for the people who doesn't know, I would like to introduce about the iPS technology. With the iPS technology, we can generate human iPS from patients with genetic background. These iPS possess unlimited proliferation capacity and ability to differentiate into almost any kind of organ-specific cell type. This has enabled the generation of disease-relevant tissues from patients in a dish. On the other hand, recent technologies such as zinc finger nucleases or transcription activator like effector nucleases, and particularly clustered regularly interspaced short palindromic repeats, also known as CRISPR system, have enabled the versatile genome engineering of different mammalian cell types, including human pluripotent stem cells. These technologies offers the opportunity to introduce, modify, or correct mutations and to generate genetically matched isogenic control lines, which would help us to establish definitive genotype to phenotypic links. Zinc finger nucleases, TALENs, and CRISPR systems create a double-stranded breaks on the human genomic DNA at specified locus. Upon double-stranded breaks in the cell repairs this double-strand break via one of its two repair mechanisms. The quick and dirty non-homologous end-joining repair pathway is error-prone and often results in nucleotide insertions or deletions, which could ultimately result in loss of protein function, that is functional lockout. If a homologous DNA repair template is provided, the double-stranded breaks can be repaired via homology-directed repair pathway, whereby the desired base pairs from the synthetic donor template are inserted in the desired sequence, that is knock-in. Each genome modifications by zinc finger nucleases or TALENs require specific design of protein modules for a specific project, which makes use of these projects very difficult in normal laboratories. In contrast, CRISPR-Cas9 system requires single universal nuclease and specific design of gRNA, which makes to use of CRISPR system is very easy and affordable to any lab. Because of differences in CRISPR system and zinc finger nucleases and TALENs, considering the nuclease design and assembly, CRISPR-Cas system has become the main tool for genome engineering projects around the world. What is CRISPR-Cas system? CRISPR-Cas9 is a bacterial adaptive immune system that protects the bacteria from invading viruses by selecting the specific sequences and degrading the viral sequences. The same mechanism has been harnessed to modify the mammalian genome. Based on the presence of CRISPR proteins, the CRISPR system has been divided into two subclasses, class 1 and class 2. Class 2 system is the one which has been validated for their genome modifying features because of the system having one single effector protein. And the CRISPR-Cas systems has further subdivided into different classes basing on their requirement of FAM or basing on the requirement of nucleic acid substrate, whether it is a DNA or RNA. So there are different types of Cas proteins, Cas9, Cas12, Cas14, and Cas13. And all these proteins has been validated for their genome modifications. And Cas9 is the first and most commonly used Cas protein, which has been using to make gene knockouts, gene corrections, or conversion of one nucleotide to other nucleotide. And also Cas9 has been using to modulate the gene expression by activating the gene or repressing the gene or epigenetically modifying the genome. All these modifications has been validated in different organisms ranging from Drosophila to plants, and also different cell lines such as cancerous cell lines or primary cell lines. Nevertheless, genome engineering in human iPSCs has been facing a challenges because of their intrinsic characteristics. Generally, the stem cells are very sensitive and very culture dependent. If there is any changes in culture dependent, they tend to change their chromosomal abnormalities. And also the stem cells to grow as a single cells has very limited or very less efficiency of single cell cloning. And they are having ability to spontaneously differentiate in cultures. So because of these challenges, it has genome engineering in iPSCs is taking more time and more resources. Because of this challenges, the progress towards realizing the full potential of CRISPR editing in iPSCs is very limited and very slow. So next few slides, I would like to share the assured process of genome engineering in iPSCs, which needs limited amount of time and less resources, and which is very specific for the design modification. Here, I summarize the main and important steps in the assured genome engineering pipeline to generate high quality knockout or knock-in iPSCs with integrity. It means with no off targets as well as without chromosomal abnormalities. Optimization of different steps such as designing, delivery of CRISPR components, or single cell cloning requires optimization of these steps are important to achieve high editing efficiency. Besides, adopting automation in making single cell clones has further improved the single cell clones quality and their integrity. And also this process increase our genome engineering process. So for successful genome engineering, it is very important to plan and design the experiment before even starting the experiments in the laboratory. There are three important steps, three important things to design to have the most time and cost efficient approach to obtain the desired cell line successfully. The one thing is selecting the desired editing. There are different kinds of genome modifications you can do in stem cells, such as knockouts or knock-ins or gene regulations. Depending on the type of modification and also depending of the type of cell line, you can choose the delivery options to deliver the CRISPR components inside the cell. There are different kinds of delivery options available, such as plasmids or viruses or mRNA or protein. You can deliver Cas9 using these different approaches. Once you decide the delivery, once you choose the delivery components, the next important thing to assess the editing outcomes. For this, there are different kinds of approaches to assess the editing outcomes, and depending on your requirement, and you can choose the best and suitable approach to have the most time and cost efficient approach. And I will describe in detail about the PCR genotyping and the inference of CRISPR edit for selected modifications. And here, I would like to go through with the pipeline or the selection and the plan of the projects which I do, which I select. For example, to make a knockout modifications, I selected CRISPR components as a synthetic gRNA and a protein. And to deliver into the cell, I choose the electroporation. And for assessing the editing efficiency, I choose the IC indel analysis for the smaller indels, for larger deletions, PCR genotyping. And for SNP insertions, I choose the IC knock-in analysis. For larger knock-ins, I choose the PCR genotyping. Because these are the strategies for the specific modifications, which are most cost and time efficient strategy. And then in the next slides, I will explain in detail how I design and how I plan these experiments for the specific modifications. So the optimization of designing. Designing a gRNA is one of the important and the most crucial factor to have the successful gene editing in iPS project. Because the gRNA, the best design and selection of gRNA will influence the high editing efficiency and low off-target efficiency. So before designing, the first thing you need to do is a collection of particular sequence. So gene sequence. These things you can collect the sequence information by NCBI or Ensemble, and then you can annotate the gene by using Benchling online tool or DNA software. So what the annotation means, you need to select the start codons and also position of exons and position of introns. Once you annotate a gene, depending on the type of knockouts, whether it is a complete functional knockout by targeting all isoforms or knocking out the specific isoform, or deletion of exon or deletion of complete gene, depending on the type of modification, you can select the online tools to design the gRNA. There are ample number of gRNA design tools, but I choose the Synthago to make a functional knockouts because it's a very simple. You can select the gene name and then insert into the software by selecting the type of species, and then it will provide the top gRNAs which targets the all isoforms. In other hand, if you want to make a simple knockout by selecting specific sequence of your interest, or if you want to delete the complete gene or complete exons, you can select the particular sequence and place into the CRISPR-Cas9 online tool, and it will design the gRNAs which targets the particular sequence. So both these tools will design the gRNAs. Among these and provide the list of gRNAs. Among the list, you can select the top gRNAs which having the high on-target activity with minimal off-target effects, and also consider the gRNAs with a minimal off-targets with mismatches, 3 or less mismatches, so that it will having the low off-target score. And then there are other considerations to select the gRNAs to make a knockouts. In this case, having the gRNA with ART at 4th position of NGG to create a indels in a way, duplication of ART next to the ART in the genome, which leads to the frame shift indels. There are other gRNAs which is having the microhomologies, which activates the microhomology dependent repair. This further leads to the deletion of sequences between the microhomologies. Depending on the sequence, it will create the type of indels, which might be frame shift or in-frame indels. Besides, there are other softwares where you can predict the outcome of indels and their percentage of indels using the inDelphi or forecast software. And there are other considerations to select the gRNA to make a knockout. Choose the gRNA which cuts the splice donor's exon site to alter the mRNA splicing. And choose the gRNA which is not too close to the start codon, which might activate the downstream alternative start codon. And also avoid the gRNA which cuts the splice acceptor site, which might lead to the exon skipping and expression of your protein. After selecting and designing the gRNAs, you need to optimize the PCR products. For this, design the primers and then optimize PCR conditions and then do the sequencing and annotate the sequencing to not have any SNPs which might affect the gRNA activity. So once you design and select the gRNAs, you can synthesize by using the IDT or Syntago or any other commercial gRNA synthesis companies. And then choose that gRNAs which is having the chemical modifications, because this gRNAs is having the higher stability, increased stability, and the less protection from the high protection from the exon nucleases, and has been shown for improved editing efficiency and also with less off targets compared to plasmid or viral delivery, where you don't have the opportunity to modify the gRNAs. And after synthesis, you need to deliver your CRISPR components into the cells. And for this, you need a Cas9 protein. I choose the Cas9 protein and also electroporation method. Because electroporation has been shown in my hands and also in other labs, is having higher efficiency in iPSCs compared to plasmids or compared to other transfection methods such as chemical transfections. And for example, to make a knockout, you can simply use one single gRNA with a Cas9, or to make a deletions, you can use the 2 gRNAs and form a RNP complex with the Cas9. And once you make the RNP complexes, depending on your modifications, you can deliver into the cells using the electroporation. I use Cas9 protein because it has been shown for higher target activity, less off targets, and no integration to the genome, and is having the minimal cytotoxicity. And once you deliver into the cells, you need to know how efficient your editing approach. To do that, I analyzed the pool after 72 hours. And for specific modification, we can choose the specific editing efficiency assessing method. For example, knockout, I choose the IC analysis because it is a very simple one, a simple bioinformatics tool, which is calculating the percentage of indels and the type of indels by deconvoluting the Sanger sequencing inputs from the control and edited sample. And also analysis shows you the background noise in the edited sample compared to the wild type sequence. And also it will calculate the percentage of indels and the type of indels. And for larger deletions, you can simply run the gel and see that shift in the band sizes. And after selecting the desired methods to deliver and the desired method to assess the editing outcomes, and you need to perform the single cell cloning. And basing on the editing efficiency after 72 hours in the pool, you can select the number of single cell clones and then perform the knockout analysis by doing the same single Sanger sequencing followed by ICA for the knockouts. And for the larger deletions, you can simply run the PCR followed by gel run. And then once you selected the clones, you can validate by the Western blot for the functional knockouts. I would like to go through with the one simple making a knockout experiment in the iPSCs. For this, I selected a gRNA using the guidelines which I described earlier. In this case, I choose a gRNA with a microhomolosis, which might make a deletions of 8 base pairs. Once I selected the gRNA, I synthesize using the chemical. I use the chemical guide RNAs, and then transfected into the cells using the Cas9 protein as an RMP, and analyze the pool after 72 hours. And then it will analysis showed that 65% of indels in iPSCs. After analysis, I perform the single cell cloning followed by IC indel analysis for the knockout efficiency. And I choose the clones which is having the 100% knockout efficiency after the IC analysis, and then will perform for the Western blot and showed for the clones with a complete knockout. Among these two clones is having the 8 base pair deletions. Next, I would like to go through with the knock-in analysis, design for the knock-ins, which is very similar to the previous design, with the only difference with the primer design and validation of sequence before the gRNA design. And once you sequence validated, you need to validate it for the mutation of your interest to be changed, or the sequence where you wanted to knock in the larger deletions. Make sure that there is no SNPs which might affect the gRNA activity. Once you select the gene sequence, you paste into the CRISPR-Cas9 sequence, and then select the gRNA, which is having the in-target activity, low off-target effects, and also select the gRNAs which is having the SNP-specific gRNAs. And once you selected the top high target and low off-target gRNAs, you also need to consider the some considerations for the kind of modifications. For example, to make a homozygous knock-ins, you can select the gRNA which cuts very close to the intended mutation. If you want to make a heterozygous knock-ins, select the gRNA which cuts the 10 base pairs away from the intended mutation. So yeah, depending on the zygosity, you can select the gRNAs, respective gRNAs. Once you design the gRNAs, and you need to design the donor templates for the smaller SNPs, SSODN ones, and for larger knock-ins, large SSODN or donor plasmid. I will give you why SSODN is important and how you can design the SSODN. SSODN1 has been shown for a higher efficiency when compared to plasmids or double-stranded DNAs. And my choice is asymmetric. There are different types of designs. My choice is asymmetric SSODN with a PAM mutations. So asymmetric means with two different homology arms, shorter homology with a CRISPR sequence, larger homology with a PAM sequence. And then you can insert the mutation of interest and PAM mutation to avoid the recutting of the desired knock-in, and also make sure if there is any gain or loss of restriction sites, which might be useful for the screening. And for larger knock-ins, you can design the longer SSODNs with the homology arms with the transgene in between. And the longer SSODNs were expensive, and sometimes it's hard to synthesize. Alternatively, you can use the plasmids as a donor template, but the plasmids consider that it's having the less efficiency. Once you design, you synthesize using the IDT for the larger SSODNs, and then deliver into the cells using similar approach, using the Cas proteins, only with addition of donor template. For smaller SSODN, for larger ones, larger donors. And after delivery, you can analyze the pool for the smaller knock-ins, IC knock-in analysis, which is similar to IC indel analysis. Only difference with the providing the donor template for the analysis. For larger knock-ins, you can use the PCR gel run. And I would like to give you one example of correction of disease-specific heterozygous SNP in iPSCs. In this case, I designed a gRNA which is specific to the SNP sequence. And then I designed the SSODM similar to the guidelines which I shown you. After I design, I synthesize the guide RNAs and the ODNs and deliver into the cells and analyze the pool for the knock-in using the ICA knock-in analysis. And you see there is a 99% of knock-in after my analysis. And after that, I perform the single cell cloning and the screening using the similar approach for the one which I use for the pool analysis by using the ICA knock-in. And among the clones, 6 clones is having the homozygous SNP, which is a T here, and compared to the control sample, which is having the heterozygous A and T. So with this, I can conclude that by designing the specific experiments, you can make the knockouts or knock-in within the limited time with high efficiency. And the most important factor is the planning the experiment for the high success rate, designing and selection of gRNAs and donor templates for high specificity, and selecting the delivery method for high editing efficiency, and quantifying the editing efficiency for the cost and time effective approaches. And besides automating the automation of single cell procedure for the high quality cells with no chromosomal abnormalities and within a limited time and the faster time. And I would like to thank Sebastian for the opportunity to work with him and colleagues for continuous support and in suggestions, and especially for Sandra for doing all editing experiments in iPSCs. And watch out for the editing in iPSCs, automation of editing, and more information for the single cell cloning automation by Katia. And thank you.
Thanks very much, Narasimha. Hi, everyone. My name is Katja, and as Alex already mentioned, I'm a research scientist and in charge of everything related to stem cells at IOTA Sciences. In the next 15 minutes, I would like to show you how our single-cell cloning platform can advance your genome engineering projects. So I'll briefly discuss common pitfalls in traditional cloning methods. I will then continue with an introduction to our cloning platform and show you how it can be incorporated and simplify CRISPR-Cas9 genome editing projects. There are various common methods for single-cell cloning, including FACS sorting, limiting dilution into well plates, and manual colony picking after low-density cell plating. Unfortunately, all of these are usually labor-intense, time-consuming, and expensive in regards to reagent usage. In addition, they lack reliable verification of monoclonality and carry a substantial risk of selecting and propagating non-clonal cells. Especially if we think of a 96 well plate, it is almost impossible to see a single cell due to the optical edge effects. So for this reason, researchers often blindly assume that a compact or densely packed round colony, as the one which is shown here, was derived from a single cell. However, that is often not the case, as is exemplified in the next image of the exact same colony. So as you can see, in reality, the round colony is mosaic and was derived from at least 2 different cells, of which one carried a green and one a red fluorescent tag. To address the challenges and bottlenecks in traditional single-cell cloning workflows, IOTA Sciences cloning platform utilizes a novel type of miniature fluid-walled cell culture chambers, which are collectively referred to as grids. So a grid, as shown here, is comprised of 256 individual culture chambers, which is roughly equivalent to 3 96 well plates. The chambers are made by using fluid shaping technology. They consist of either cell culture media or matrix coating, are fully separated from each other and overlaid by a translucent liquid, namely an immiscible fluorocarbon. So here in this image, the chambers contain red dye for the sole purpose of visualization. And this makes it easy to see how small their footprint actually is. So each chamber comprises an area of around 3.2 square millimeters and utilizes less than 1 microliter of medium to culture single cells into colonies. So let's have a closer look at these grid chambers. So on the left now, you can see images of 2 individual such chambers, which contain a single cell each. These images were taken directly after cell plating using a 4x objective in bright field mode. And even if the cell is close to the rim of the chamber, like in this bottom image here, it is still easily visible as there are no optical edge effects which are otherwise common with conventional well plates. Both adherent and suspension cells can be dispensed and cultured in these grid chambers. So on the right now, you can see some exemplary pictures of single-cell derived clonal colonies, which have been maintained in the grid chambers for 7 days. So the top image shows a clonal colony, which was derived from a single adherent cell. And then in the bottom image down here, you can see a clonal suspension culture. So these grids form the foundation of a streamlined single-cell cloning workflow, which is automated on IOTA Sciences' cloning platform. And this platform consists of the isocell, seen here on the left, and the isohub, displayed here on the right. So the isocell is a small, compact, and standalone system. It creates grids and takes over all of the related pipetting work for the grid chambers. The isohub features an Olympus microscope and works as an information and control hub. So it aids the identification of grid chambers containing single cells, as well as the tracking of colony outgrowth. And both systems automatically synchronize data with each other through a wireless connection, which ensures a seamless and controlled workflow. So let's have a more detailed look at how the cloning platform can be incorporated and simplify a typical application in the stem cell field, such as a CRISPR-Cas9 genome editing workflow. Narasimha has already given us some expert insight into the first steps of a typical genome editing workflow. So after transfection and cell recovery, one usually ends up with a heterogeneous pool of cells carrying different gene edits, as well as cells which haven't been edited at all. And the only way to isolate a homogeneous cell population is through clonal expansion from a single cell. The whole single cell cloning process, which comprises isolation of single cells, regular media changes, identification of individual colonies in either 96 wells or flat bottom dishes, as well as harvesting and expansion of colonies, is usually pretty time-consuming and labor-intense. So especially as traditional cloning approaches do not allow the user to reliably verify that they have started this process with a single cell, there's a high risk of selecting and propagating non-clonal cells, as the one which is shown here. So this in turn has a detrimental effect on the last step of the genome editing workflow, the downstream analysis. Because if one ends up with some kind of mosaic population at the end of the single cell cloning workflow, he or she has to go back and then start this whole cloning process from the beginning. So to avoid a lengthy cloning process, it is best to get it right from the start. IOTA Sciences cloning platform now not only simplifies the most intensive steps of the whole single-cell cloning workflow, but it also allows the user to verify monoclonality before cells grow into colonies. Within just 1 week, we can get from a single cell to a true clonal colony, which then can be passed on to this further downstream analysis. So here now you can see in a bit more detail how this is done. The cloning workflow starts with the isocell fabricating grids and then automatically dispensing cells into individual grid chambers. And after the cell plating, the isohub is then used to identify chambers which contain a single cell. And thereby, the isohub automatically guides the user through an entire grid. And once the user visually identifies a chamber which contains a single cell, he or she can select this particular chamber simply by pressing a button on the controller down here, which comes as part of the isohub. And this then activates that chamber and unlocks it for further downstream applications, such as feeding, which is done with the isocell, or tracking of colony growth, which is performed with the help of the isohub. Once the clonal colonies then are big enough, they can be automatically harvested by the isocell and then transferred to conventional culture plates. The automatic wireless data transfer between the isohub and the isocell ensures that both systems always contain the same information regarding the location and numbers of activated chambers and what has been done to them. So for example, which of the chambers have been fed or which ones have already been extracted. Before now coming to the end, I would like to briefly show you an application example where the cloning platform was integrated in a genome editing workflow, a CRISPR-Cas9 mediated GFP to BFP conversion assay. So this was performed in a human iPSC line which carried 2 copies of the eGFP gene in the AAVS1 locus, so it was homozygous for GFP. And then here now, you can see part of the eGFP and BFP sequences. 2 nucleotide changes, namely the C here as well as the G here, are sufficient to convert GFP into BFP. So we used a guide RNA targeting the eGFP sequence and HDR donor oligos, which contained these 2 nucleotide changes. 3 days after transfection, cells were seeded into the grid chambers, and chambers containing a single cell were identified with the isohub and then tracked over a week. So here now, you can see an example image of an attached single iPS cell in a whole grid chamber. 7 days later, it has grown into a clonal colony, as is exemplified here. So in total, we assessed 3 isocell dishes containing grids, and on average, 86% of single cells in the grid chambers did grow into clonal colonies. The clonal colonies were then evaluated via fluorescent imaging. And the results for the individual dishes, as well as the mean of these, are shown in this stacked bar graph here. So on average, around 36% of clonal colonies contained cells which only expressed BFP, as is shown here in blue. And this suggested that both copies of the eGFP gene had been converted. Approximately 15% only expressed GFP, displayed in green, which indicated that these cells hadn't been edited. And 48% expressed neither GFP nor BFP, shown in gray, consistent with indel events. And in the case of editing only one allele, cells of respective colonies expressed both GFP and BFP simultaneously. And this overall occurred in less than 1%, which is displayed here in yellow. So we then also assessed the editing efficiencies in the transfected bulk culture, so basically in the pool of cells which was used for single cell cloning using FACS. And here you can see that before transfections, all of the iPSCs were GFP positive. And then after transfection, the bulk culture contained cells which expressed just BFP, then just GFP, BFP and GFP simultaneously, or neither of the fluorescent proteins. And now the stacked bar graph reveals that the percentages of edited cells in this bulk culture very nicely match the proportion of single-cell derived edited clonal colonies in the grid chambers, which I've shown you here before. So this application example now already brings us to the take-home messages for today. I have introduced to you new types of small cell culture chambers, which are collectively referred to as grids. And compared to traditional cloning methods, the grid chambers come with several features beneficial for single-cell cloning, as is listed in this table here. In particular, I would like to highlight the unique optics they provide. So single cells can be seen easily, directly after cell plating, and even if they're close to the edges of the chamber. In addition, the chamber's small footprint reduces the media consumption by around 300 times compared to reagent usage when following traditional cloning approaches, which usually utilize 96 well plates or flat bottom dishes. And the grids form the foundation of our synergistic cloning platform, which comprises the IsoCell and the IsoHub, which automate and streamline the cloning workflow for grids. So while the IsoCell facilitates grid fabrication, cell plating, and colony maintenance, the IsoHub eases identification of single cells in the grid chambers, as well as tracking of clonal outgrowth. Thus, together, they simplify the most intensive steps of single-cell cloning, which we have very nicely seen in the aforementioned application example. I would now like to thank you for listening, and I'm very happy to take questions.
Thank you very much to both speakers. We would now go ahead and take some time for questions. And it looks like we already have quite a few. Now, in case we don't manage to go through all of them today, we will make sure to get back to you in writing shortly after the webinar via email. So please feel free to continue submitting your question. Now, the first question to Narasimha, have you ever encountered a situation where CRISPR-Cas9 could not be applied successfully? Did you ever come across a scenario where, for example, you were thinking about using a zinc finger or talon in replacement of Cas proteins in your genome engineering projects?
Yeah, thanks, Alex. It was a good question. Yeah, we had a couple of projects where we couldn't find the successful gRNAs which can make a cuts on a particular gene. And in that case, we tried to look for other genes where we can cut far away from the target site to eliminate the gene and then replace with the intended sequence. So otherwise, we haven't so far tried zinc finger or talon nucleases, but that would be another alternative options to genes which we cannot target by CRISPR-Cas9. Or also in the CRISPR-Cas9, there is also another options. You can try, for example, base editing or prime editing if you want to correct some mutations which you can't correct by CRISPR and the HDR pathway. So yeah, that would be, yeah, we had a couple of examples where we couldn't target with the CRISPR, and we were thinking of going into other alternative options.
Right. Okay. And just a follow-up question on that: is it like a sequence-specific aspect that the CRISPR can't work well? Is it something like GC rich content,
Yeah.
loci, for example?
Yeah, I think there could be two different possibilities, because sometimes if there is so much GC content, it might not be perfect site for the cutting site. And also there are some genes which might not be open with a chromatin. For example, there are like heterochromatin, like which are more close and more not accessible for the Cas protein. Yeah, in that cases, maybe we are not able to successfully target the site.
Okay. Katja, did you have any experience on that aspect? Have you ever looked into using zinc fingers or talons over Cas proteins for specific projects?
No, luckily so far I only had projects that worked with the Cas9 and the normal CRISPR system.
Okay.
Yeah, I mean, I would like to add that, most of the projects, at least 1 in 10 projects, 9 in 10 projects works with CRISPR-Cas9. There are 1 or 2 which are mostly tough to target, but still you can use other CRISPR alternatives like cutting other, faraway sites, as I said before.
Okay, so what you're really saying is it's not worth necessarily looking back into zinc and talents because the work associated might just be too large and you prefer going for another Cas protein version.
I think so. I think, yeah, according to my experience and also from the literature, and yeah, it's
great.
is becoming a more household technique to target the genome.
Right. Okay. We've got a next question relating to grid fabrication. Which reagents are used for grid fabrication and how are grids fabricated? Katja, do you want to take that one?
Mm-hmm. Sure. The grid fabrication requires like a thin aqueous layer, which could be either made out of coating matrices or just normal cell culture medium, and that is overlaid by like a thin translucent bioinert fluorocarbon, which we call FC40 star, which we then also supply to our customers. In general, I would say like that most of the common cell culture medias are compatible with the grid fabrication. And so the isocell itself, she then uses, the isocell uses a jet of FC40 star and basically pushes this thin aqueous layer of media or coating like slowly to the sides. So the chambers are like completely like surrounded and overlaid by the fluorocarbon.
Okay. There's another question, technical, on the isocell. What is the protocol for colony extraction? I think the question here is, are they being extracted as single cells or are they being extracted as clumps?
So the harvesting actually follows some kind of like conventional cell harvesting workflow. So if you have some like adherent cells, the isocell first removes the media from the chambers, then washes them twice with PBS, and then adds a dissociation reagent of the user's choice. So for example, with stem cells, we usually use EEE. And then after a short incubation time, then there's another program which is called extract. And there the isocell then basically takes up all of the media in the chamber, oh no, sorry, not the media, the EEE in the chamber with the cells and transfers this to some kind of PCR tube strips, which contain media already. And then this can be manually transferred to, for example, 96 well plates or 24 well plates.
Okay, so it sounds like it follows a rather traditional harvesting workflow, just kind of automated from the user perspective.
Which is, though, one thing which I think is quite interesting, which like a lot of people ask, because we quite often get the question if the cells then are kind of harvested as single cells. And even that we use EEE, which you would expect to get a single cell suspension, the isocell somehow is quite, it's somehow quite gentle on the cells so that they still come out in clumps often. So that's really like beneficial then for the outburst. So if you transfer them, you already have little like colonies usually in your 96 well plates, and then they actually do really well after that.
Okay, great, thank you very much. There's a question referring to the guide RNA design. Narasim, I think that might be a question regarding your presentation. It says in the presentation, you mentioned different software tools. What's the difference between them? And I think that the question refers to the guide RNA design softwares that you mentioned in your slides.
Yeah, so I mentioned actually in my talk, there are 2 different softwares. I mean, of course, there are like a bunch of online designing tools, but I prefer these 2 because, for example, if I want to make just functional knockout without a preference of any sequential targeting, I can just give the genome in Synthego designing tool, and then it will design the gRNAs, which are the mostly targeting whole isoforms, like to make it a complete functional knockout. In this case, we cannot really, select the specific sequence you wanted to target. Whereas other one, which is CRISPR-Tefar, in this online tool, you can provide sequence of your interest, and then you can specifically provide the sequence, and the software will design the gRNAs, which is particular for the specific sequence.
Right.
And so that you know that exactly where you are targeting and what you are targeting. In this way, this is like, there is a bit kind of flexibility to target a sequence of your interest. And also in the CRISPR effort, there are different options of Cas9 and gRNAs for different Cas9s. For example, if you want to select gRNAs for the Cas12 or Cas13, you can also choose that tab, like, okay, I wanted to select gRNA for Cas13 or Cas9, so that, it can design for the specific of your interest. Whereas with Syntago, you cannot do that kind of manual designing.
Right, I see. Okay, thank you very much for that, Nara. Do you analyze predicted off-target loci to confirm gRNA specificity? So I think you mentioned that obviously you want to try and have a high target specificity, but there might be scenarios where I reckon you might not have perfect scores and you might have a certain off-target prediction. Are you actually going in and check this in cells?
Yeah, exactly. It's a good question, actually. That is one of the most important concern for the field, to really know that what is the off-target effects of a particular gRNA, or what are the off-target effects in the cell line which you are deriving. So our design procedure contains really, we try to avoid the gRNAs which is having the most off-target effects. So these things you can, when you design your gRNA, you can already check what are the off-target effects. Like, you can choose the genes which are having the mismatches for the particular gRNA. And then you know that these are the off-targets for your particular gRNA. And then using our protocol, like, we are trying to avoid off-target effects by using the synthetic gRNAs or Cas proteins, which were shown that they have less off-target effects. And for example, in a case where you can't find the very good gRNA and which is having the potential off-target effects, those things you can also use the different Cas9 versions, which is called, like, there is a hi-fi Cas9, which has been shown for the low off-target effects when compared to the Cas9. In this case, if you can also use the hi-fi Cas9, or to really confirm that the cell line which you are generated doesn't have any off-target effects, you can select the predicted off-targets, like at least for top 5 coding region off-targets, and then do the Sanger sequencing, and then check for the indel analysis if there is any cuttings happening in these off-target sites.
Right.
So far with our protocol, we haven't seen any off-target sites, at least in the cell lines which we generated.
Okay, but you're just doing this in the case that your gRNA score is maybe not the highest, or you have like a certain predicted off-target, and it's then when you go after, essentially.
Yeah, the gRNAs which we select really, like, there are really like minimal off-target effects, like mostly like 20 to 30 off-targets. And if that is the case, you really don't find in your cell lines. But for some cases, you find like gRNAs which is having like more than 200 off-target effects.
I see.
So in this case, you can select top 5 off-targets and then do amplicon sequencing or simply Sanger sequencing followed by indel analysis. So yeah.
Right. Okay. There's a question concerning the delivery of reagents into cells regarding transfections. How can we improve transfection efficiency of stem cells? I think there's a question going on to both of you. Do you have any most critical parameters that you find most critical to increase transfection efficiency in your hands?
Yeah, so I can go first because in my experience, in the beginning during optimizations, we checked with lipofectamine-based transfections and also electroporation, and also using different electroporator machines. And during this experiment, at least I could say that in our case, using electroporation has a high efficiency, high editing efficiency compared to lipofectamine. And yeah, I mean, and there are papers show that even lipofectamine also works in a similar fashion. And it's only thing is you need to optimize what is the right condition and what is the right amount of lipofectamine for your particular cell line. And theoretically, yes, you can do all the transfection methods.
Okay, Katja, do you have something to share, some critical parameters?
Yes, so I think what happened to me before was that when I tried to do and introduce a specific SNP and had to design an SSODN as like a template, we had to optimize the concentration of this template because it seemed like when we added a too high concentration of that it led to a lot of cell death after transfection. for some reason. So by reducing the concentration of that, we get it to work.
Okay, that's an interesting aspect, because there's a question here on we experience a high rate of cell death after transfection of iPSCs. How can this be avoided? You just mentioned one parameter in your experience was the concentration of the SSODN, so the repair template. I mean, I guess culture health is obvious. I mean, are you doing anything in particular before you go into transfection when it comes to the culture, for example? Are you changing medias out, or are you using different reagents such as clona up front before the transfections, things like that?
Yeah, so yeah, it's a very important thing to have very good culture, like very good cultures with a healthy culture conditions. And to have very good culture conditions, it's always recommended to grow your cultures in the best possible conditions with the best media and the single cell boosters such as clonar or Revitacell. And all of them works in the similar way, and sometimes it depending on the cell type also. So depending on the cell type, you can optimize which culture conditions are good for your cultures, growing as a single cells and also for the transfections. So and normally what we do is whatever the cell cultures were growing before, we always try to adapt to the cultures which we use, like stem flakes, for example, and also with the clonar conditions before transfection. Especially before transfection, we split the cells in a way to have the next before transfection, in a way having all the cells in the proliferative state or S phase condition. It has been shown that when the cells are in growth phase, there is a higher percentage of transfection as well as editing efficiency. So yeah, that's why it's a good thing is to optimize your culture conditions before you go for the transfections. Right.
Okay. And do you see a large variety of transfection protocols between different pluripotent stem cell lines? I mean, do you need to optimize it for every new line, so to say, or do you have, so to say, a working horse that works across a bunch of cell lines in your hands?
So far, considering the transfections, we always use the same conditions, like same voltage and same pulses. We don't change anything regarding the electroporation. But when it comes to the culture conditions, sometimes some cell lines will not like the one media which was regular uses. So there are some cells, they are really particular for some culture conditions. In this case, maybe you really need to use the same culture conditions, not the one which I use, but yeah, it's for the cells which they like mostly.
Right.
For example, some cell lines really likes the MTSR media with a clonar. Some cells really like the just Temmax screw with a Revitacell. So there are some cells really very particular about the culture conditions. In that case, we can't really do much.
Yeah, okay. Okay. There's another question, technical question on the single cell cloning aspect. What is the volume? I think you might have mentioned it, Katja. What is the volume that get dispensed in the individual grid chambers?
Right. Yeah, that is like 200 nanoliters, which the isocell dispenses in each of the chambers across this entire grid. And then, yeah, we usually leave them in there for like a few days before we then basically top up. So in the end, you have like less than 1 microliter in total. Then you actually culture them into colonies.
Right. Okay. And the further top up, does that somehow affect the optical aspects of the grid chambers then?
No, it's still, because the volume is still so low, they is still quite easily visible even if it's just a single cell. But usually by the time we top up, you will see quite like tiny colonies already. Even if they spread out a bit, it's still they're still very well visible.
Okay, thank you very much. I think we might have time for one more question now. And as I mentioned earlier, if we didn't manage to pick up your question today during the webinar, we will make sure that we get back to you after the webinar via email. So last question, which coatings and medias are compatible with the grids and the isocell cloning workflow?
Okay, so basically most of the like commonly used coatings are compatible. So we had very good results with laminine 521, then recombinant metronectin, as well as Geldrex. There is unfortunately like definitely one coating, which is Synthemax, which unfortunately is not compatible with the grid replication. But otherwise, it seems to work really well. And concerning media, I think that most of the media which are commonly used in, for example, especially the stem cell field, they're fine. However, because we see best results with like laminin, Stemflex, Dona, we usually recommend to use that, or definitely use like a kind of nutrient-rich medium aside cloning. Even if your normal maintenance culture maybe is in like just normal something like E8, we then advise customers normally to switch just for the single cell cloning process to this like richer medium, and then you can easily transfer back again without any effect on like the potency or genomic stability of the stem cells.
Right. Okay. Well, thank you very much for this. With this, I would like to conclude today's webinar. Many thanks to our speakers for the very informative presentations and following question and answer discussion. We hope you found it useful and hope to see you again next time. Bye for now.
Bye.
Thank you. Bye.